I am Sarah Kostick. I'm a postdoctoral associate at the University of Minnesota in the apple breeding program, and I've been with the program for about 2 years now.
What do you think is like the most interesting part of what you do, or what piques your curiosity?
Everything is a puzzle, and everything is basically prediction, right? So when I design crosses, like what parents I'm going to put together, it's there's science there, but there's also art, right? So we know what these parents look like. We know some of the genetics of these parents, but we don't necessarily know what types of offspring they're going to produce. So I'm like, well, we're going to put these together and we're going to see how it goes. And a lot of times it's not great. It doesn't end up being a good idea. And then sometimes you're like, wow, that was kind of an out of the way cross there that I wasn't sure was gonna give us anything, but it really gives us something interesting. So it's the balancing of that art and that science that I find is really fascinating in plant breeding.
When you're doing something like that, like how long does something like that take?
Like planning crosses and determining how we're gonna approach it. So a breeding program has a cycle that we do the cycle every year, right? And so we're making crosses every year, and me as well as members of our team start sitting down, and probably, let's say after Christmas is that typically when we start sitting down and going, okay, going over our field notes of evaluation, what did we notice in some of the parental combinations that was working, and what did we notice that was not working, and going from there. And so it takes a couple months for us to put together those lists. And then you're constrained by biology. You can have a plan, and then the weather and whatever blooms, and then you're like, well, we're gonna have to rejigger our plan a little bit. But yeah, it takes a little bit. So basically, with a lot of fruit quality traits that are important to our perception of flavor, etc., they're what we call highly quantitative, meaning that you don't have one gene that's controlling them. You have multiple small effect genes throughout the genome that have an effect. But having a marker at any one given gene is really not going to give us an idea of how high is the sugar going to be in this potential individual. And so what genome-wide prediction does is that it basically says, okay, we have all of these genetic markers throughout the genome, and we don't necessarily know what each marker, how it's associated with a given trait. But what we can do is we can utilize those and put them into a linear model or some sort of model to predict performance. And so when we have this breeding population, we use all of those markers and we build a model. We have to train that model and develop it. So we have to be able to have a population that we know both the information about the trait as well as the genes. We build that model, and then we predict performance in another population, or those breeding populations that we're really interested in.
Do you know if certain markers are going to have a bigger influence than
others? Initially, no, we don't. But what you do through building this model is that you estimate marker effects. And so there are going to be some markers through the linear regression process that we identify as, oh, these have larger effects. Now, what does that actually mean? It just means that that region has a larger effect on the overall trait. So, for example, for acidity, because I know that there are 2 loci that are involved for sure, and other genes as well, I predicted those. And then if I went in and I looked at the marker effect, markers at the MA locus, which is on chromosome 16, and then the MA3, which is on chromosome 8, those have larger effects than the other random markers throughout the genome. So yes.
Okay. Mm-hmm. Are there other traits other than sugar that you do this for?
So we're looking at, right now, genome-wide prediction is still within the testing phase of whether or not this is worth our money to put in and time to put into an overall breeding program. But yeah, so we're looking at sugars, or soluble solids content, which is our estimation of sugars, MA, or malic acid, which is our estimation of acidity. And then we have multiple texture traits. So a couple of firmness measurements, as well as our estimation of crispness. And then I have done color just to see, can we do it? Color is one of those traits that as you know, there's one region that's really important, but there's still variation within there. So we can predict color as well.
I think I had a little bit of a harder time seeing where the research is right now with like firmness and crispness. Can you talk a little bit about that?
So texture is a little bit more complex. There's been quite a bit of work on texture because as a consumer, the first thing that you experience when you bite into a piece of fruit is the texture. You're not going to really care about flavor if it's mushy. And so there's been a lot of work on texture, primarily firmness. There's quite a few regions of the genome that are involved. There's been some that have been identified, PG1, which I think it's on chromosome 10. That's involved, and there's other genes that are involved. We still just don't fully have a complete understanding of texture in general, partially because texture is experienced through multiple different traits, and it's a combination of the interaction between those. So firmness is really important. How hard is it for you to get your teeth into the fruit, but also crispness. So how do those cells burst? And so you need both of those to really experience texture. And crispness is really complex, and I don't think we fully figured it out.
When did this type of work start? Like roughly, like was this like 5 years ago?
Yeah, so this is really genome wide prediction started in animal breeding, actually. So when you have a dairy, you're trying to predict milk performance, right? When you use a certain bull, that's how they utilized it initially. And so this has also been in the last 20 years or so been a point of focus in agronomic systems, so corn, soybean, wheat, etc. In apple, it's been in the last, I would say, 5 to 10 years that this has really become a point of interest. We're behind a lot of the other crops just by nature of the complexity of apple from a perennial production system, as well as just it's genetically pretty complex. But I would say in the last 5 to 10 years.
Is that due to the extreme heterozygosity, or is that other pieces as well?
Partly it's heterozygosity, partly it's apples are self-incompatible. Part of that is just apple has a very long juvenility, and so our generation times are really, really long. And so in agronomic systems, you're cycling pretty quickly. In apple, we have to wait for fruit to evaluate, and so it takes a long time. You can only speed that up so much, biology in general.
What did you mean by self-incompatibility?
In order to get fruit, right? In order to get to get seed and therefore fruit, you need a genetically distinct individual that is a source of pollen. And so that's what I mean by that, is that it can't self for the most part.
Okay, got it. And then are there other fruits that, like, are apples one of the main fruits that this work is being done on, or is this
genome wide prediction stuff that actually, from the fruit crop perspective, the most advanced groups are in strawberry, actually. So they're doing a lot of work. The university, down in Florida, the breeding program has done a lot of work on genome wide prediction, and they're actually utilizing it in their breeding program. So apple's definitely not at the forefront. I think strawberry is the best fruit crop example, which should make sense. It's a lot shorter generation times, etc.
I was wondering if you might be willing to share a little bit about like how you got into this or what your background is.
Yeah, no, that's a great question, and I love answering it. So we'll go back to high school. I was really interested in biology and just the natural sciences, but I didn't really know what you could do outside of medicine. And so initially I'm like, well, I'll become a doctor. I did my undergrad in biological sciences, and then I realized medicine really not the path for me. I actually had an internship at the University of Minnesota Landscape Arboretum and Horticultural Research Center, which is when I first got introduced to plant breeding. That really opened the door of like, oh, you can be a scientist, you can do a lot of really cool genetics work, and have a direct application in agriculture. We all have to eat, right? And we all love to eat. So I did my master's here at the University of Minnesota, and then I went out and did my PhD at Washington State University in the apple breeding program out there. Was really introduced to apple production on a very large scale. Apple's a really cool crop. Not a lot of apple breeders out there, but you need to be able to have plant breeders in all crops. And so job security pretty solid there.
Thank you so much for your time.
No problem. No problem.
I really appreciate it. This is really helpful. And just like, I think it provides context because sometimes looking at studies can be overwhelming.
Very jargon heavy often, and we all do it. So just, yeah, I try not to, but we get stuck in our little jargon and it gets confusing.
Well, you did a really great job of explaining it and demystifying it. So thank you.
Yeah, no problem.